from Ploof. I am a first year student at Lone Star College, and today I will be talking about the effect of light or dark incubation on bacteria, trying to find different isolates from soil. So, antibiosis is a biological relationship where one microorganism prevents growth of another. Soil bacteria such as bacillus can actually produce antibiotics to use it as a competitive strategy. And so for my conditions of isolation, I used light and dark exposure to see if it can affect the growth of certain bacteria. So with light exposure, it favors photosynthetic bacteria, and with dark exposure, it mimics underground environments, so it favors heterotrophic bacteria. These conditions can shape the diversity and composition of isolated bacterial populations, and it increases the likelihood of identifying antibiotic-producing stains. So this led me to ask, how does the light or dark conditions of incubation affect the isolation of bacteria colonies from two different soil samples? So my objective was to test the effect of the presence or absence of light during incubation on the number of colony forming units, also known as CFUs, of the bacteria isolated. So this is my methodology, which has quite a few steps. My first step was to get the soil. So I went to the beach at Galveston, Texas. I was near the seawall, and I had dug around 2 inches into the sand to get my collection. And when I collected the soil samples, I recorded information such as the pH levels, the temperature of the air, the temperature of the soil, and the GPS coordinates of the findings. And then I also got some soil from a rose bush in my backyard in Dayton. Next. So then I did serial dilution. So basically I made a slurry of my soil samples, which combined 1 gram of the soil with 900 microliters of PBS solution. And then I made a mixture of 100 microliters of the soil slurry and mixed it with 900 microliters of deionized water. And then I had made 4 dilutions. The first dilution was actually not diluted at all. It was just the first mixture by itself. And then with each dilution, the dilution factor would be multiplied by 10. So then I made my library plates, which you can see right here. There's different, with the dilution factors going lower, there's less bacteria, which is what we were hoping for. However, some of them have some plates that just kind of go all over the place. And so I had made 16 library plates. 1 was for each dilution, being dilution 1, 2, 3, and 4. And then these were all 1 soil sample and 1 factor. So the top one was soil sample 1, light. And then it would be soil sample 2, light, soil sample 1, dark, and soil sample 2, dark. The CFUs were counted, and I used that information to create an equation to see how many CFUs were on the plate per gram. So there was actually a bit of an accident with the library and patch plates. These are my patch plates. I was able to make all 4. 1 for soil sample 1 light, soil sample 2 light, and so on. And each patch plate had 16 different isolates. And these were isolates that I had gotten from my library plates and had tried to get a single colony by itself. That way I could further analyze the individual CFUs. However, when I went to do my preliminary screening, one of my plates had been lost in the lab, so we don't know where that plate was. So instead of having 64 possible isolates to test, I had to go down to only 11. And so that had definitely hindered my research, but I still got some results. So for the preliminary screening, I had tested it against S. epidermidis and B. subtilis. And all 11 isolates were tested with the bacteria to see if they show any signs of antibiosis towards common bacteria. If you can see up here, there is a slightly clear area around the bacteria. You can see it better in these 2. And that is showing that it is producing antibacterial against S. epidermidis and B. subtilis. And so with that, we went on to the next step, which was the escape safe relative testing. So instead of just using 2 bacteria, we used 9 instead. And so I did the same process. All the plates were basically the same with still the 11 isolates. And so I used the bacteria such as E. coli and E. raffinosis to soil the plates and then transfer the isolates that I had made from streak plates into the plates with the bacteria on it to see if there were positive results from those bacteria as well. And so my analysis is as follows. On average, there were more bacterial colonies in the dark conditions compared to light, which you can see with the averaging for light soil sample plates to be around 95, and then the averaging for dark soil sample plates was around 160. So this did help with my hypothesis that dark conditions were going to have a difference with the light conditions. However, I would like to do more tests because I had a low amount of testing that I was able to do. There were 32 isolates that had tested positive for antibiosis. However, this was out of 99 isolates. So that was the 11 isolates times 9 with each bacteria. So out of those 99, only 32 had tested positive. However, 23 of those were dark condition isolates. So these were the isolates that had no light exposure. And then 9 of those were light condition isolates. So these were my results for the 9 bacteria. These are all of the bacteria tested, and then at the top it has soil sample, the number, and then the dark or light. So 1 is going to be the Galveston sand, and 2 is going to be the rosebush dirt. And so S. epidermidis and B. subtilis were the most common ones to have positive results against. However, there were 2 that did not have any results. That was A. bailey and P. putida. And the reason for that is because these plates were tested at 28 degrees Celsius. However, A. bailey and P. putida actually are, they grow best at 37 degrees Celsius. And with the Galveston sand, a lot of these soil samples actually had positive results against bacteria that wasn't being tested positive in the rosebush, such as E. raffinosis. So 82% of my tested positive isolates were against B. subtilis. And then only 9%, that one from Galveston, was tested positive against the E. raffinosis, and then 27 was with E. coli. This is just a basic measuring of where each bacteria stands. So my conclusion is that with dark versus light, on average, dark had more CFUs and bacterial growth than the light isolates. However, I would like to see more in having different soil samples and having more replicates. With the soil versus the sand, on average, the rosebush dirt, soil sample 2, had more bacterial growth. And then with the escape bacteria, only 32% of the isolates had tested positive at all. However, I would like to have more replicates as I only had 1. So obviously more tests need to be done because of human error, because of some issues that happened in the lab. But these are the results that I had with what I had been able to do. These are my works cited. Thank you.